and welcome to today's Salon webinar, part of our ongoing series to explore research, careers, and the technologies shaping the future of dementia science. I'm Adam Smith, and I'm delighted to be hosting this fascinating session on exploring image-based machine learning. And I have to start, but first of all, apologise that we're a little bit late in getting going today. And also highlight that we're having a few technical problems, not most because I failed to mute my phone. I'm muted now. We're having a few technical problems, but please bear with us. We'll hopefully get through it. But let me set the scene for this topic today. Artificial intelligence and machine learning are no longer science fiction. They're powerful tools being used right now to advance dementia research. From brain scans to biomarker prediction, image-based machine learning is opening up new possibilities in diagnosis, monitoring, and understanding disease progression. But it also raises questions. How accurate is it? Can it outperform human clinicians? And what are the risks of biases? And most importantly, how can we ensure that technology benefits the people who need it the most? We're joined today by two outstanding speakers who are working at the intersection of medical imaging, AI, and dementia. We have Dr. Sindhuja Tirumalai Govindarajan, who is a postdoctoral researcher at the University, I know, I was so close, of Pennsylvania, working in AI and biomedical imaging lab, or the IBIL, where they are applying machine learning and advanced image processing to study neurodegeneration. We also have Dr. Prashanth Gouda, who is a medical researcher with a clinical and academic background across top institutions in India and Oman. And with over 40 publications, his current work at the National University of Science and Technology, Muscat, focuses on integration of AI in clinical diagnostics. They're each going to take a turn at presenting for 10 minutes each. And then, as most of you know who joined us before, we'll have a chance at any point during the conversation for you to post your own questions and share your own experiences on this topic. And then we'll all come together for a chat at the end. As you can see, that's one of our technical problems. We seem to have letterboxed Prashanth, but hopefully we'll sort that out while Sindhuja's very kindly agreed to present first. Over to you, Sindhuja.
Hello, everyone, and it's nice to see the interest in my area of research, and I'm excited to learn more from Prashanth's work as well. I will be sharing my screen because I have some introductory slides that can help us with the discussions later on. While I get that ready, I'd like to warn you that I'm recovering from a cold, so if I sound a bit nasal, I apologize. Okay, so the topic today is image-based machine learning, and as Adam mentioned, I am a postdoctoral research fellow at the University of Pennsylvania. I'm also an Alzheimer's Association Research Fellowship awardee, and my work relates to application of machine learning and AI towards modifiable risk factors to dementia. But today, I'm hoping to give you a quick primer on neuroimaging because I don't recall a salon session on imaging specifically in dementia. Why do we even have to care about imaging? Because it allows us to, in vivo, identify whether a person has a neurological condition, whether it's a particular type of condition, and also to rule out other kinds of diseases that could be like stroke, or TBI, or other kinds of injury that could also result in cognitive impairment that presents as dementia. With the in vivo identification, we're also able to tell with imaging what type of dementia it could be. Is it of vascular origin? In combination with biomarkers, of course, we're able to tell if it's AD or frontotemporal dementia. In vivo imaging allows us to do it with a little bit more information of what's happening in the brain. It also allows us to monitor progression of disease within the person. And usually this is done through types of imaging. I'll group them as structural imaging and functional imaging, which I'll talk about later. Structural imaging allows us to study the patterns and the extent to which the brain has degenerated and caused atrophy. Think volume loss or tissue loss, which results in shrinkage. We're able to visualize it on structural images. We're also able to tell the degree of damage due to vascular reasons, vascular factors, whether they are lesions in the white matter or microbleeds and other sequelae. We're also able to measure the integrity of microstructure itself using some advanced methods. For functional imaging, we can measure activity of the brain while the participant is, thinking about something or performing a task. We're able to measure how different parts of the brain are active with metabolism. We can measure deposition of amyloid beta plaques across the brain. We're also able to measure neurofibrillary tangles, or what we call the tau tangles, through in vivo imaging. So the structural imaging, I'm giving you some examples. On the top are what are called T1 weighted structural MRI, and on the bottom are CT MRI. They offer different information, but what we're seeing here is the staging of atrophy, starting from a relatively intact brain to a lot of atrophy, where you see larger gaps and smaller brain tissue, larger ventricles, which means more CSF, which means the brain tissue has shrunk. With the vascular damage or lesions, I'm showing you an example called FLAIR imaging, which allows us to visualize these spots. They're also called white matter hyperintensities. And clinicians have a scale that they rank participants on or patients on based on how much and how distributed these lesions are. This is called the Fasekas rating scale. I mentioned microstructure. Microstructure, essentially, what I mean by that is we're able to measure the axons, which form the white matter. And these are the pathways that connect neuronal cell body to neuronal cell body. So think of your brain as a massive electrical system. DTI can be used to measure if the wires are damaged, connecting the different switches. We also have functional imaging. On the top is resting state functional MRI, which is measured based on oxygen consumption in different parts of the brain when a person is, just thinking about nothing in general, no active task inside the MRI scanner. And on the bottom is FDG PET. It's a glucose-based ligand, which allows us to see that in a normal participant, there's glucose consumption pretty much throughout the brain. It reduces in MCI. You can see in AD there's hypometabolism, which means the brain regions are not as effective in energy utilization. And this we're able to all measure in vivo. More relevant to Alzheimer's disease are the amyloid plaques, dementia in general, the tau tangles as well. We can use radioligands that are specific for binding with these proteins of interest. And we can see that in normal older adult, with the lower amyloid and tau, the maps are almost very low uniformly. In another older adult who has higher amyloid and tau, we have increased absorption of the tau tracer and somewhat increased absorption of the amyloid tracer as well in certain regions. Whereas in an AD patient, you can see substantially increased absorption or binding of the ligands for both amyloid and tau. So this is just an introduction of the type of very commonly talked about modalities in imaging for dementia research. And I just want to also highlight, conventionally, we know that these directions are expected. I'm talking about research from 10, 20 years ago. We know our signatures or patterns of expected changes in the brain by comparing a group of controls and a group of cases or patients in this case. We have histograms of the different features. We try to find a separation point and we say, oh yeah, this particular signature is clearly different between the groups. And we take this average and then we say, yep, this signature is for the case, this median signature is for the controls. However, what these types of analyses ignore are the fact that patients don't all have the same disease pattern, nor do the controls. There's substantial inter-individual heterogeneity. Also, there could be very subtle changes, early changes that we don't yet detect. And what happens if we want to measure how a particular person progresses along the disease continuum? Case control studies cannot handle that. And further complicating all of that is that we just saw there were so many different imaging modalities which offer complementary information on what happens in the brain. We also have multiple pathologies or comorbid conditions that occur. And currently, not even currently, these type of analyses from 10, 20 years ago could not handle them except by separating the clean data sets of participants with only one type of pathology, participants with only one set of images, and things like that. However, machine learning can help us move beyond that and place patients along a continuum. So it allows us to summarize a disease pattern and assign a score to a participant, which can be placed along, let's say, this panel or this three-dimensional space, where each dimension could be a particular pathology, each could be a particular way of disease progression, and it allows us to see patients all along there and measure them as they progress further in the continuum. So this allows us to improve or assist in diagnosis and monitoring of participants and progression of disease. It also allows us the discovery of new markers. We can integrate multiple data sets together rather than relying on, one at a time or what is possible in one particular institution or one particular study or one particular scanner. We're able to now discover new markers that can be applicable across multiple domains. We're also able to detect much more subtle patterns, which we would not have been able to do otherwise. What does this mean? This means we can do personalized assessment and we can monitor participants. This allows us to stratify participants by risk. We can say, oh yeah, these participants are at high risk. Let's see if we can enroll only the high-risk participants in a clinical trial and test how the drugs work on that group versus the low-risk group. It also allows us to compare on an individual level whether they got better or they got worse over time. I hope you're all with me so far. Adam, please interrupt me if something happens. Okay. To illustrate what I just talked about, I have a pop quiz that I love sharing. I'm not able to see anyone's chats or video, so I would love to see your feedback or your inputs later after my presentation. I have 3 participants here. These are gray matter density maps, which means they're atrophied or not atrophied. You can see all of them have lower gray matter density in some regions. On the right are white matter hyperintensity maps, which are lesions of vascular origin. All 3 of them have it. Now, I want to see if we can match these 3 images to these 3 subjects. We have one 73-year-old female participant with hypertension, obesity, and type 2 diabetes. One 65-year-old male participant with only hypertension and nothing else. And one 58-year-old male participant with obesity and type 2 diabetes, but not hypertension. I will wait for your answers. I will show you what we can do with machine learning now. So this is based on my paper that came out last month. We are able to use machine learning. This is a classification-based algorithm to assign a number which summarizes. Think of a pattern that is associated with a particular disease. We can scan a participant and say, oh yeah, this person has a severe expression of this pattern or not so severe expression of this pattern. And using that, we generate these numbers. So this is the 58-year-old male participant who had obesity and type 2 diabetes, representing very high values of those corresponding markers. Here, this participant with only hypertension had a very high value for the hypertension marker, but not so high for the other 2 markers. This female participant, who was 73 years old, expressed very strongly the effects of hypertension and type 2 diabetes, but not obesity, even though they had a very high BMI. But this is just an example of what machine learning can do. And I want to share more examples which are by no means comprehensive, but are easy to access. So by using AI and machine learning on structural MRI markers, we can create a panel Think of it as a panel like blood work panel. You go to the doctor, you get a blood test done. You can measure your blood glucose, you can measure your blood lipid levels, and you end up with a panel which tells you how high or low they are, what requires attention, what you're doing very well on. What we're suggesting with machine learning is you go get an MRI scan, and out comes a panel which tells you which signatures are very strong and worrisome, which signatures are okay. In this case, the example is a cognitively normal participant who's 83 years old. They don't have AD, so they don't express the AD marker. They have hyperlipidemia, and along the longitudinal follow-up, they had hypertension. So they had very high values of those, but they are somewhat relatively okay with the other markers. A different AI algorithm performed by a student in the lab, she used generative AI for personalized reference estimation. What this means is, imagine you were able to look at a brain and rewind or restore what it would have looked like had it not aged, had it not had any risk factor. That's what she did. So she created a counterfactual, which is personal to each brain, and calculated the deviations. So these are the areas where this person's brain deviated from how they would have been when they were completely healthy and young. This is made possible through generative AI. A different student on the same participant performed some longitudinal modeling to show how they're differing across the different regions. So if we only have the baseline values, the black lines are where these regions are supposed to go in time from baseline. These are based on global estimates based on the whole data set. However, using a deep kernel was able to recreate or formulate the trajectory, which was much close to where the participant was in these data points. And here we can see this participant has above normal, like better than predicted hippocampal volumes in longitudinally, somewhat close to expected values, maybe a little poor on the thalamus, almost directly overlapping expected trajectory for lateral ventricles. So this is one example. And the second example here, very similar. This is a participant with AD, who's 75 years old, so younger than our previous participant. They have a very high spare AD score, and they were likely a smoker who did not report it as such. Here are the personalized deviations. There's a very strong right temporal parietal deviation that we can observe. Again, the personal counterfactual assumes the recreated image is without any AD or aging effect. So this is how their brain would have looked if they did not have that. Using the deep kernel regression for the longitudinal trajectory making, we see that the participant was not always 80, even though this snapshot was from an 80 time point. They started out with mild cognitive impairment and progressed over time. If we only had the baseline information and the population level trajectory estimation, the black would be the line, where we see here the participants' hippocampal volumes are somewhat overlapping with what's expected at the population level. But in the thalamus and even in the lateral ventricles, we can see that the data points are much farther away than the population estimate. And the student's model is actually approaching that estimate. These are just some examples. And other strengths of machine learning algorithms, of course, here's some integration of not just MRI, but MRI and genetic data to identify subtypes of dementia from a large data set. This gives us information that cannot be achieved just by looking at images. Here's another model, it's called Sustain, which is very widely adapted to other imaging modalities, but was formulated in structural MRI, which allows for simultaneous subtyping, one or two, as well as staging, so how people progress along those stages. These are by no means comprehensive of what machine learning and AI can do, but just quick examples to kind of give you a highlight. Some of the common cautionary notes, I'm sure we'll talk more about it later, but some cautionary notes are that no one model rules them all. This is not Lord of the Rings. The choice of imaging features and the training model highly depend on the question that we're asking from that particular data. Imaging features tend to be numerous. We have more predictors in general than participants, which is the outcomes. So we need large data sets in order to be able to do justice to the prediction with the machine learning models. And when we have more advanced models which do nonlinear fitting, they tend to overfit really, really well and give really high accuracy on the training data set, but perform poorly on external data set. The problem or the challenge of generalizability. So usually this can be countered by extensive cross-validation using multiple data sets which have different distributions of the patient and control populations. But the use of multiple datasets also comes with technological sources of variability. MRI scanners introduce intensity variations based on who the make on the model was, what type of sequence it was. And there are ways to counter them in order to not mix the technological variability with the biological variability. Some of the methods are harmonization or combat GAM. You can look up more about it and we can talk about it. And also we could opt for deep learning, which tends to learn, supervised deep learning methods tend to learn only the biological source of variability because they're very good at nonlinear fitting. So I hope I was able to share some thoughts on how image-based machine learning can change the way we approach dementia research. I shared some examples as well of how they can identify patterns that humans might miss and some common challenges. With this, I stop sharing and hand the stage over to Adam and Prashanth.
That was fascinating, really good, and super comments from everybody as well, talking about, oh, it's been a really great introduction, which I wholeheartedly agree with. I'm interested in something you mentioned earlier on, because we're currently working on a plan for a conference at the moment, which is talking a lot about biomarkers, and you hear a lot about kind of machine learning in that biomarker prediction space. And one of the things that we've tried to integrate into this program is looking across the spectrum of biomarkers, because there's been so much progression in every area, whether that's neuroimaging and blood biomarkers and everything from eye tracking and gait analysis to cognitive testing and other things. Is you touched earlier on about the ability to integrate other data. Is there potential for machine learning there, not just to kind of, read a million scans to be able to be good at predicting, but also to take on board other pieces of information to improve that modeling?
Of course, it's not a blanket yes. There are nuances to it. What I mean by that is if we have different types of data, we want to be cautious on how many predictors we are providing the model from a particular data type and how salient there are. There are strategies around how machine learning can, how we can build models to select relevant data while minimizing redundancy. It's called MRMR. There are ways to put it in, but yes, machine learning and AI models can certainly integrate multiple modalities of data. There are some great papers on it, which I hope we can discuss.
Has anybody got any questions for Sindhuja while we're waiting? We've lost, unfortunately, we seem to have had a problem and have lost Prashanth. Hopefully Prashanth's on his way back, but if anybody has any questions, we'll take that. Are these open source data platforms like PyTorch and TensorFlow and Nil Learning, are they making things easier, Sindhuja?
They are, I believe they are a lot easier now than they used to be, as you can imagine. I do want to caution anyone starting to be patient or maybe find someone who has done it to guide you to the right source for learning, because the number of tutorials and the number of people promising to teach you AI have also exploded along with the use of AI. There
And
may be
I
some
suppose, where are we at with the publication of papers on these? I mean, the validation and what's, because reproducibility, I'm guessing, is a bit of a challenge on some of these studies, because unless you're going to give them absolutely everything, so much of the work is unique that it you can't. I mean, you could train another AI with exactly the same data, but would you get the same results?
That's a
Because
great
of course
question.
would it learn
I,
differently?
there are several different aspects to what you just asked me. So let me start with the reproducibility aspect of it. Journals, which publish methods and publish new research on it, are highly, highly advocating for reproducibility assessments to be done, for transparency about the method use, as well as for open source packages to be available that anyone can download and try on their own data. For example, my paper, I had to publish with a generated DOI, like an identifier, so that even if I update my package, that original version doesn't change and it's always referenceable. I published that along with the data. I cannot share the data. I guess that's common for several groups working on this. If the data and the data transfer agreements that you have forbid the sharing of source data, then all you can do is publish the numbers you found with a coded identifier with no other information. However, data availability statements, along with clear indications of where the source data came from, are almost mandatory in most high impact journals, at least from what I know. Your question on whether if you take the same data and the same model, will you get the same result? Do you, ideally, that should be the case. One of the a paper that my advisor wrote in 2019 for neuroimage, I think it has a very great example. A lot of us talk about cross validation of data sets, and making sure you test your model on an external data set to see if it's generalizable. But no one is cross-validating for the number of models a student or postdoc tried until they got to the model that performed reasonably well. So there is definitely an element where it may not be plug and play, given the data could have been processed differently, the data source could be, you know, there's a different combination of participants in one versus the other. There may be nuances to it, but the advantage of using such large data sets, however, is that you're able to measure something beyond the noise of heterogeneity that comes from a few missing subjects. Yeah.
I'm with you. When we get into, there was a question about diversity. How diverse, how much diversity is the data that they're being trained with?
I would like to clarify what type of diversity we're talking about. Are we talking about clinical
I'm
diversity or
guessing.
demographic diversity?
Well, I would guess both gender, ethnicity, kind of those particular elements, I think, often come to mind when we look at this, because we know that study participation tends to be kind of white middle class people overall, particularly in clinical trials. Is whether we're collecting that they've clarified they meant ethnicity.
I'd like to share a great bit of study from maybe 10 years at this point, which is a lot of the functional MRI studies, which looked at how the brain behaves when it's resting, how the brain behaves when it's doing a memory task or an attention task. A lot of them were done at universities, and a lot of the results and findings were relevant only to college students who needed the money from participating in those studies. not to the general population, not to general people of 20 to 25 years of age or anything like that. This was a published study and it made waves when it came out. So, yes, there is a risk that comes from using historical data because historically there weren't targeted efforts to maximize the population representation within a data set. The UK Biobank, for example, is a group of very healthy people. And the neuroimaging part of it are, I'm going to say, the ultra healthy people who manage to go to the scanner and get their brain scanned. So the diversity goes, the need for diversity is acknowledged with recent, until recent efforts going towards maximizing it, at least. I want to add the element of diversity of also clinical characteristics. And I say this because studies for ADNI, for example, which is the biggest known, most popular study for Alzheimer's dementia, they pre-screened people for vascular conditions at baseline, which may have been rewarded in later phases, but in the earliest phases, they were people with no hypertension or diabetes. And that's kind of telling us a very useful, very important, but a pie, a section of the pie, not the whole information. So diversity of state data sets is important. There are efforts towards maximizing that. Some of the tests we do in our lab is see if we can find comparable effect sizes within subgroups of our data. By that, I mean we cannot change how the data were collected 20 years ago, but we can measure whether our models are performing equally well in the 5% of the sample, which is not quintessentially middle class white. Yeah.
Well, that's, yeah, I mean, I suppose that's a great advantage of technology, is being able to make up for those differences and test your models in those different populations of people as well. I'm interested just to take a slightly different chain of direction for anybody who's watching this, who's a kind of an early career researcher working in neuroimaging, or is kind of how do you find your way into this career path? Because it feels like you don't have to come from that imaging background. You could have come from a computer science background, or you could have come from so many different areas to apply this. And often at Dementia Researcher, one of our aims is to encourage people who don't necessarily work in Alzheimer's, or young people particularly, to think about applying their talents to this challenge that is Alzheimer's and dementia's diseases. What brought you to this career?
That's a great question. I've been working with neuroimaging. I come from a biomedical engineering background, and I trained in aspects of neuroimaging that include acquisition, pre-processing the data. I scanned people in the past, but now I work in a computational lab which does machine learning and AI, and most people in the lab have never scanned a person before. It's the reason I say this is, as you said, yes, we have people from an applied math background, people from computer science. We have a neuroscientist who works with us who came from a neuropsychiatry background. So there are many ways to take your career trajectory, if that makes if that gives hope. I
Absolutely.
will optimistically add, I will very optimistically add that it takes all of us to do good research. It takes different flavors of researchers and scientists to ask different types of questions in order to move science forward. With
And do
regards
you
to
find
how, I'm not sure I can answer the question of how in a very general manner. Perhaps if someone is interested and they want to chat with me after, they can reach out and we can brainstorm some options.
we have the fundamental element to our community if people are looking in
Yes.
in that space? I think it is. I mean, as you know, somebody who's works around this area, not specifically in any particular field of research, I've always found the whole element of machine learning to be a little bit of a gray area. Like, it happens, but it feels like that's some magic that goes on somewhere in the background. Practically, is this, I don't know if anybody, this may be a dumb question for other people, for those that are in the audience who are far more aware of this, but are these, are these big computers in your university that you use, or are you using resources from other places? Are these accessible to other people? Are this computing power and this available to people in the world, or do you have to be at a particular place to do this?
That's a fantastic question. I, I appreciate you for asking these questions on behalf of early career researchers. I'm using resources at my institution, and I've interacted with people in smaller institutions having constraints on their modeling abilities because of the resources as well. And I don't have a quick hopeful message. I haven't done the reading on this, on what options are available, but I do know that more and more these days you are able to request computational access. Perhaps not, I want to run a model for 3 months that takes 3 months to train, but what is it called? DPUK, Dementia Platform UK, is that correct?
That's right,
I believe
Dementia's
you're
platform.
able. That's right. You're able to perform computation on the platform, correct?
I believe I believe that you can do that. You can do analysis in the DPUK dashboard and
Or
request that from
perhaps,
there.
yeah, perhaps someone working with that can add to this conversation, because I don't want to give false hope or false answers, really.
It's interesting. I
There's
mean,
some.
it's kind of, I think there's an increased awareness of this I mean, it's not the same. I fully appreciate that, but with kind of some of the AI tools that we're being given these days, through the obvious big ones we've all heard of, and increasingly you're seeing that they're broadening out the capabilities of these ready-made tools to everybody, where you can feed it 50 pictures and say, do things with these pictures, that you could start to see that this really, researchers that don't have skills in machine learning or with AI that some of these tools are starting to be built in such a way where they're ready made for you to ask, give things and ask questions of it, where it makes your research can be informed by these tools, even if it's not in an applied way, or the complexity that, of course, you do. We've got a question. Let's take this question from Jack, who, I can't flag it on the screen because it came into the chat. Let me
May
move
I read
it.
it?
I'm going to move it from the chat into here, and then I can flag it. I can spotlight it on the screen. There we go. That's the question. There will be two primary factors to consider. How do you pass the effects of pre-existing diversity in the brain, resulting in age, ethnicity, and development? Secondly, how do you recruit a sufficient sample from each pool of participants? So this is back to our making points on our question about diversity from before. I'm interested. I mean, obviously, we know that diversity is that it's everything about diversity is not just genetically or physiologically, biologically, but that from the background and upbringing and changes in diet and changes in culture in different places, do you see those differences in the brain? I mean, are those differences present in the brain beyond what you'd see, say, when you scanned your average American? Do you see differences in the brains biologically?
Yes.
Sorry, I'm asking dumb question.
Yeah, well, it's no, no. You're so good at asking questions with multiple aspects, but it's not a yes
Sorry,
or no in
I'm
the end.
it's
I
because
want
I'm
to
poor at asking
say
questions.
beyond genetic makeup from birth and ethnicity, there are elements of it. Again, what does ethnicity mean? Does it mean the color of your skin or the way you were raised? Did you have more exposure to the outdoors or did you have more exposure to industrial pollution? All of these do have an effect on the brain. And as Jack very rightly mentioned, how can you possibly pre-correct for these confounding factors and make sure you have the right sample or the right number of participants for each group? I think realistically, we've moved a little bit past that in machine learning. And I say this because there are different, if you want to go at an intro to AI or intro to these kind of methods, they'll tell you there are two groups of methods. One is a supervised machine learning or AI method, and the other group is unsupervised machine learning method, and they both have their uses. When you do something supervised, you tell the program, hey, this is the only label I care about. I don't care about whether they are from Australia or from Papua New Guinea. I only care whether they have the symptomology or not. And the machine learning algorithm or the AI, which is more nonlinear, will like deep learning stuff, which is more nonlinear, will bend through the data and find only parts of the brain or the signatures that most reflect changes because of that condition. Whereas if you do unsupervised, some of the options with unsupervised learning have been to subtype disease, which is you tell the program, hey, there are some clusters in here, there are some groups of people in here. I don't know what they are. Can you find them? And it goes through the groups and tries to put them together. And then you learn more about groups of symptomology that occur together, or signatures that look somewhat similar across a group of participants. So there are ways to post hoc investigate whether whatever the AI found has enough of a strength across these subgroups. There are also ways to find, if I found 5 subtypes, is subtype 5 more related to autism, or I think that's the example that was mentioned, like a neurodevelopmental disorder, than subtype 1. So there are ways to ask that and learn more from what the model produces. Does that answer your question somewhat?
I think it does, absolutely. Jack made another point as well about, obviously, Jack's must be working in this field. Perhaps we should have had you jump in, Jack. You could have joined us as our second speaker. Private repositories on GitHub can be used while your programs are in development. This can allow you to have an effective version control as you improve the code, decrease runtime, better memory, CPU usage until the code is ready to publish alongside the final paper. At which point the code can be made public for further improvements, which good points. Given our technical problems and everything else, we did ask, so using a popular AI high street tool, we've trained one of these. We've trained an instance of ChatGPT using all of our blogs. We have over 3,000, nearly, not almost, we have almost 3,000 pages of content on the Dementia Researcher website. That's over 400 blogs. We've been publishing a podcast for 5 years now, and that's gone on every topic you can imagine. And all of those have been transcribed, and we've trained an AI model on all of the content from our website. So not from the internet, just from the stuff that we trust, because we published it. And when I usually wrap these shows up by asking a question, which is what would be the top 10 tips on any given topic based on what it knows from our website. And these are the top 10 tips for researchers interested in image-based machine learning that our tool gave us. Let's see if Sindhuja agrees with these. Number 1, it says, start simple. Learn basic Python and experiment with pre-annotated data sets. Would you agree with that?
100%. There's no reason to start with the most advanced model. Start
Right.
simple. I would advocate for start with a simple question as well, not just a simple model. Learn data, think of it as data science rather than the next ChatGPT. Yeah.
Well, and also as well, if you're in the UK, the National Centre for Research Methods run a whole bunch of free or very low cost coding skill workshops for particularly for early career researchers or those that are new to coding, which I know even qualitative researchers have been using to learn how to integrate those. Number 2 was don't just throw data at a model. Understand the theory behind your algorithm.
Did I write this?
No, no, you didn't, but it's great. It sounds
Yeah.
this is learning from our website. Number 3, always validate on a separate data set. Overfitting is real and misleading.
That's a
It's number
fantastic advice. It's a caution thrown at everyone who wants to try um, fitting advanced
num-
models to data.
Number 4, collaborate with clinicians and computer scientists. It's a team sport, which you already said exactly the same thing. That one of the strengths of your team is that diversity that's there. Number 5, use public data sets like ADNI or UK Biobank to explore and prototype. Again, or Dementias Platform UK, which we've put links to ADNI and DP UK in there. It's almost like we knew this was coming. Number 6, read papers beyond your own field. Innovation often comes from cross-pollination. So don't just read about neuroimaging ML papers. Go and look at machine learning that's been applied in, I don't know, in motorsport and Formula One, or because Race Against Dementia, their latest fellowship application wanted there to be an AI machine learning element to that because they're keen on stuff like that. Number
I
7,
would
document.
say, can
Sorry,
I
go
add
ahead.
number 6? I would say number 6, don't let reading hold you from doing. That tends to happen a lot to me.
Well, I have to admit, I'm one of those people. I would rather read. I've watched every YouTube video on photography and very rarely get out and take photographs. I need to do more of them. Number 7, document everything. Reproducibility is a major factor. We've talked about that already. Number 8, look into explainable AI. It helps build clinical trust and improve design. Number 9 was seek ethical review early, especially when working with human health data. I know from experience, sitting on a national research ethics board, that a lot of people who volunteer on ethics committees, in the UK particularly, we do tend to attract kind of particularly a lot of older people are on there, and that doesn't make them less capable, but it does mean that you really do need to explain this. When you are applying for ethics, you might find that they're not going to be as aware as you are, so do seek that early. And last of all, number 10, stay curious and skeptical. Not every pattern is meaningful. Be rigorous. So
I like the last one likely applies to life.
yeah, I completely agree. So given that that's all drawn on resources from the Dementia Researcher website, do we pass the test?
Pass the test.
None
Well,
of those points were wrong, right?
no, they were all correct. They were all correct, and I agree with them. I'm just wondering, did we miss anything? Um, I would reiterate that no one model is going to be better than everything else. So definitely keep that in mind and work in a contextual way. If you're interested in predicting a numeric value, go for a model that does that in your data set. There's not the best model and the most cited model, but the most appropriate model for the data set and the question. I want to say I was looking forward to Prashanth's discussion with the explainable AI. Perhaps can you have him again over? another day?
Absolutely. I'm so sorry that we lost Prashanth. I will follow up with Prashanth and give him an opportunity. Maybe we can jump on one evening and just have his talk and have a chat. Absolutely. I'm really sorry we lost Prashanth, because I completely agree. Because you managed to speak beforehand, didn't you? About what he was going to present on.
Yes, there was an email conversation. Yes, we did talk about that. Yeah.
Well, thank you very much for bearing with us this afternoon through our technical gremlins. You've been an incredible audience. Thank you so much for all this discussion we've had in the chat, which is great to see. It's really lovely that you're all so enthusiastic on this topic. And also thank you to the brilliant Sindhuja for stepping up, giving a wonderful presentation, and also as well for bearing with us and keeping going when we lost our second speaker this afternoon. We will be back on this topic again. We'll get Sindhuja back when we can iron out our technical problems, and we'll hear from him again. But in the meantime, thank you very much. I'm not here next week. I'm going to be at the British Neuroscience Association Neuroscience Festival in Liverpool. If you're going to be there, do come along and say hello to me. I'll be taking headshots, and we're going to be recording a live podcast on raising your profile, and also running a couple of CB clinics. So if you're in Liverpool, do come along and say hello. And if you're not and you're staying here, next week we have Ella Moonan Howard from the Alzheimer's Society, who is one of their innovation leads, who's going to guest host. And we have two speakers, including Dr. Sam Moxon, one of our regular bloggers, who are going to talk about how they've taken something they discovered at university and created their spin-out companies, spin-out businesses from their learning, and how they're innovating using funds and partnering with organizations like Innovate UK and the Alzheimer's Society to take their discoveries and become businesses. So not just for scientists, it's also relevant for those who are involved in care and developing services or products. So if that's for you, do join us next Wednesday at 12 noon for that webinar. Otherwise, I'll see you in Liverpool. Thank you, Sindhuja, and we'll see you
Adam.
all again in the salon next time. Have a great day. Bye.
Bye.